Hello everyone. Today we are going to discuss one another topic, that is Avery MacLeod carty experiment. We already started molecular basis of inheritance chapter, and in which we already discussed about the Griffith experiment. Griffith, who was one of the scientists and who was studied on DNA is a genetic material on the basis of transforming principle. He was said that this theory was called as the transforming principle. And but different scientists, means this Avery, MacLeod and McCarthy experiment, those scientists were not convinced to that of the transforming principle, so that they used different test tube assay, and they proved that DNA is a genetic material in 1944. They were purified strain, and they were take extract of the protein, RNA and DNA and one another material. They were used extract of strain of the material, that is protein, RNA and the DNA. Then they used different three test tube. They used three test tube, and in which they used first test tube for the protein material, in which at the time of this experiment they used three digestive enzyme, in which they used protein in one of the test tube, and we know that that is protein which is the one of the chain of amino acid. In that extract of protein, they add one of the protease enzyme, and this protease enzyme, it is one of the digestive enzyme, and this digestive enzyme, protease enzyme, affect to that of the protein and break down the chain of this protein, so that in which Avery and MacLeod and McCarthy, they were said that only DNA is a transforming principle. But in a transforming principle, but in which when protease enzyme affect to that of the protein and break down to that of this chain of amino acid, so that it is not affected on the transforming principle. Means transformation जी process आहे त्या transformation process मध्ये या प्रोटीन्समुळे काहीच अफेक्ट होत नव्हता. मीन्स दे यूज हीट किल एस्ट्रेन बॅक्टेरियम और एस्ट्रेन मिक्स विथ दॅट ऑफ द आर स्ट्रेन. जे याच्यामध्ये त्यांनी दोन वेगवेगळे स्ट्रेन यूज केले. एक हीट किल एस्ट्रेन घेतला आणि एक सेपरेटली आर स्ट्रेन घेतला. आणि हे जे आर स्ट्रेन आहे, in which he was they observed that is transformation not affected. Means they take different protein, RNA and DNA for what? To see which one could transform. From R strain to S strain. Because in the Griffith experiment what happened? That is lastly काय केलं होतं ग्रिफिथने, की लास्टमध्ये त्यांनी काय केलं होतं, मिक्सचर मटेरियल घेतलं होतं. कशाचं? हीट किल एस टाईप प्लस आर स्ट्रेन, लाईव्ह आर स्ट्रेन मिक्स विथ दॅट, अँड इन विच दे वेर ऑब्झर्व दॅट द माईस वॉज डाईड. आणि ते जे डाईट झालेला माईस होता त्याच्यापासून त्याचं जे ब्लड सॅम्पल घेतलं आणि त्या ब्लड सॅम्पलचं कल्चर केल्यानंतर त्यांनी ऑब्झर्व केलं त्याच्या कॉलनीज ऑब्झर्व केल्या. त्याच्यामध्ये त्याला सगळे आर स्ट्रेन कन्वर्ट टू द एस स्ट्रेनमध्ये दिसल्या. ठीक आहे. तशाच प्रकारे ह्या तीन सायंटिस्टने इथे तीन टेस्ट ट्यूब घेऊन ती जी प्रोसेस आहे, म्हणजे आर स्ट्रेन कोणतं मटेरियल तिथे ट्रान्सफर होत आहे हे बघण्यासाठी इथे त्यांनी टेस्ट ट्यूब बेसिसचा एक्सपेरिमेंट प्रूव्ह केलेला आहे. देन they use second test tube, and they were used the extract of RNA, and in that test tube they were used RNase enzyme, and they used RNase enzyme. We know that this RNase enzyme affect to that of the RNA we know that it is one of the single chain, and in which single strand. Then this RNase, it also break down to that of the RNA, and it is also not affect to that of the transformation. They observed that the RNA it is also not RNA is not also responsible to transformation, so that they take third test tube, and in which they observed DNA was breakdown and inhibit the transformation, means in which only those material that is called as the DNA was affected on that of the transformation. DNAs which is digest the DNase enzyme, it is one of the digestive enzyme. It affect on the DNA, and it also affect to that of the transformation. ट्रान्सफॉर्मेशन वर अफेक्ट दाखवला आणि ट्रान्सफॉर्मेशन अफेक्ट म्हणजे इथे एक्स्ट्रॅक्ट घेतला कशाचा, दिस DNA विच इज टेक्स फ्रॉम हीट किल्ड estrine. Heat killed estrine plus means mixed with that of the DNase enzyme, and they were observed, that is the transformation is also inhibited. Okay, and on that things, this Avery, MacLeod and McCarthy proved that DNA is act as a genetic material. But different scientists, again scientists were not convinced to that of the theory. So that again Hershey and Chase experiment was also proved DNA is a genetic material, or it is one of the transforming material or transforming principle.